Okay, can go on. So, as said, my name is Mirko Coggi, and today I'm talking about pangenomics, and in particular on how a graph-based approach will improve genome analysis in the next few years. So before starting, I want to leave you some notes about me. So my journey in this field of genomics started during my bachelor and master degree in biomedical engineering, where I discovered to have a great interest in computational genomics, and also in creating optimized solution which leverage the hardware acceleration, and also, and in particular, in pangenomics, which is the study of multiple genomes at the same time, like analyzing the genome of a population, for example. And this drove me here today because currently I'm a PhD student here at Politecnico di Milano. And as my colleague Guido said before, I contributed to the foundation of the startup Genogra, whose vision matches my research interest, which I will explain in this talk. So, talking about computational genomics, everybody knows that this field has gained a great importance in different contexts, ranging from the personalized medicine to the radiomics, to the metabolomics, the live and feedstock industries, and many, many other contexts. But despite this great amount of application, we can see that the genomic pipeline is pretty much standardized, because it always starts with the taking of the DNA sample, which is then sequenced to obtain subsequences of the original genome, and then the computational task begins, because these reads needs to be assembled together to recreate the original genome, which is finally analyzed to find some specific sequences of nucleotides inside it. So until 2015, both the assembly and analysis tasks could only be supported by a linear representation of the genomes. And this led to the creation of a consensus reference sequence, which was made by merging a multitude of genomes into one single sequence by selecting for each genome site only the most frequent variation among all the possible variation for that site. And this approach introduces a great limitation, which is called reference allele bias, because the data incongruent with the referent allele is underreported. And in particular, during the read mapping process in resequences, this results in a systematic misrepresentation of the authentic sequences that build the reference. And moreover, this approach can also limit the scalability when we have to deal with a population of genomes. And in the end, this is also inefficient to represent the structural variation, so the bigger variation inside the genome. For this reason, in recent years, genome graphs has been proposed as a more efficient and a more compact way of representing the inter and intra-individual variability, because of their ability to encode variations as alternative paths inside a directed graph. The problem here is that graphs are more complex structures than sequences, and this introduces new technical challenges when we have to develop graph-based computational pipelines for the genome analysis. In particular, the main challenge regards the research of specific sequences or patterns inside a reference, which has usually been called the read mapping process, but that in case of graphs is the sequence to graph alignment process, which consists of computing the alignment of the query sequence to a path in the graph, having the maximum alignment score among all the possible paths. Well, the successful execution of this task is biased by different factors. For example, the accuracy and completeness of the results, the flexibility of the tools, and also the performance improvements on top of the alignment algorithms. So in order to investigate the trends in developing this type of tools, so sequence to graph aligners, I implemented a novel methodology for a comprehensive analysis of the state-of-the-art tools. In particular, this methodology allows for a qualitative analysis of the three factors I mentioned before, where each factor have been decomposed in a set of parameters that collected together allow for evaluating the aligners on specific data and in specific case studies. On the other side, I also implemented the first open source benchmark suite for this type of tools to quantitatively analyze the accuracy and the execution time. In particular, the benchmark suite is completely automated to test the performance of the different aligners over a pool of data, which comprises both the reference graphs and the reads to be mapped to these references. And I think it's okay. So in my first version of this work, I included 7 state-of-the-art tools, which every one of them promises to solve any critical issue. But however, I noticed 3 crucial aspects in this study, because I discovered that there isn't a solution with outstanding performance on all type of data. But different tools performs better on data with different characteristics. For example, one of the main characteristics is the linearity of the reference graph. For example, some tools go faster on more linear graphs and other tools on highly branched one. Then another fact is that these tools are slow. So there is no solution integrating hardware acceleration solution, for example. And in the end, a crucial aspect is that next generation sequencing technology allow for a faster genome sequences, which increase the production of genome data. And this allow us to create larger and larger graphs, but current solution can't handle such big data. So these highlights helping me in defining the scope of my PhD thesis, that is the creation of a definitive framework for graph-based genome analysis to solve the issue of current solutions. And in particular, I thought about a framework which is composed by 5 tasks, which starts with the graph partitioning to break the original large graphs into smaller subsections according to the branching density. Then there is the selection of the best aligner for each partition based on the characteristic, of course, of the partition. And after that, the main task is the execution of the alignment, which will be done by using a already optimized solution present in the state of the art, or with new promising algorithms that I will optimize by myself. After that, there will be a need to manage the cross-partition alignments, and in the end, the merging of the results of this hybrid alignment. In particular, the first, the third, and the 5th task can also benefit from hardware acceleration with parallel computing on GPUs with the obvious aim of speeding up the process. So currently, I'm mainly focusing on the 3rd task, so the alignment execution, because it is the more computational intensive, and also because different analysis pipeline needs different features and different fine-tuning of all the possible parameters. For this reason, I recently developed a new implementation for the GWFA algorithm, so the graph wavefront aligner algorithm, which is a really promising strategy for the sequence to graph alignment because of its outstanding performances in terms of execution time. So, however, the first implementation of this algorithm lacked the computation of the alignment tracebacks, so the precise sequence of edits needed to map the reads on the graphs. For the reason, I re-implemented this algorithm, allowing a more complete retrieval of all the alignment results, and so making it usable in real case studies. On the other side, I'm currently working on a GPU implementation of the GEDMAP aligner. GEDMAP is another interesting tool that tries to solve the problem of memory occupancy of graph structure, because as I already said, graph structure are more complex, so they need more memory to be stored inside the machines. And in particular, this tool converts the graphs into elastic degenerate strings, obviously reducing the size of the data structures, but increasing the execution time. And that's why I'm focusing on implementing another accelerated solution using GPUs to, of course, speeding up this process. So, well, creating tools, of course, can have a great impact in genome analysis, but I think it's not enough, and that's why I'm here. So I proved that the efficacy of my solution on public data available from open source databanks, but I aim to do a little bit more, because my idea is to use graph-based genome analysis pipeline in real case studies and scenarios. And that's why I'm looking for collaboration with bioinformaticians to identify new possible application of genome graphs in the most used pipelines, and also to create hand-to-hand solution that could really enhance the genomic analysis, unleashing its full potential. So with this, I have concluded. Thank you for your attention. And if you're interested in a follow-up, don't hesitate to reach me out.
Thank you, Mirko. So I will go with a quick question. And you said that sequence graph alignment is biased by accuracy, flexibility, and performance. So how did you decompose these factors to evaluate the state-of-the-art tools?
Okay, thank you. So for what concerns the accuracy and completeness of the results, the parameters that needs to be analyzed to assure that the accuracy is enough is, of course, the supporting of cycles inside the reference graphs, but also the, as I already said, the computation of the traceback, because without this feature, we are not able to use the tools in real case studies or real case scenarios. On the other side, there is also a point on the completeness of the results, and in particular on the standardization of the input and output files. And so the research is pushing the idea of adopting the GAF format for the output of the alignment and the GFA format for the input of the alignment, because these type of formats are the more complete present in the state of the art. Then, for what concerns the flexibility, two factors that are fundamental inside a tool are the possibility to have a custom scoring matrix to adapt the different tools to different scenarios, and also the possibility to have an NFI gap penalty. On the last side, the performance improvements, the most interesting solution integrated an inter-sequence parallelism, usually leveraging multi-threading on CPUs, but there is an interesting push to adopt GPU's implementation for this task, so to spread the computation of different sequences on parallel devices. Then another fundamental aspect is the use of heuristics. In alignment heuristics, like banded alignment, which reduce the compute space, and also seed and extend heuristics, which reduce the execution time by